When we talk about eukaryotic organisms, and that's a big key here, we're talking eukaryotic only, we're going to go through this regular pattern of growth, DNA duplication, and cell division that we know as the cell cycle, which is what you're looking at right here. And your cell cycle is going to have two major stages to it, and then there's going to be sub-stages within that. The big one here is interphase, okay? And if you take a look at all of this orange and sort of think of this as a clock, all right? If this clock is the single lifespan of one cell, the vast majority of a cell's lifespan is spent in interphase. The other phase is your mitotic phase, which is going to include all of this right here. That's going to include your stages of mitosis that we're going to talk about. It's also going to include cytokinesis. So, like I mentioned in the previous slide, interphase is your longest phase of the entire cell cycle. This is where your cells are going to do most of their growth, their development, and to prep for division once they're ready. Okay, so this is going to be most of their normal function, they're going to grow, and then once they get past a checkpoint here, they're going to utilize this area to really sort of prep to go into cell division next. Now, interphase has three subphases to it. You've got your gap1 phase or your G1 phase here, your synthesis phase, and your gap2. And if you look at it, the vast majority of even interphase is spent in gap1 because that's where we're talking about a new cell growing. When we go through cell division, it's going to be on the smaller side, so it's going to need time to grow, to develop. It's going to carry out its normal functions. So if it is a gland of your thyroid, it's going to be there to secrete thyroid hormones. If it is a neuron, it's going to be sending neurotransmitters and electrical impulses to communicate from your brain to the rest of your body, so on and so forth. All right. So it's going to carry out its normal function there. We get to your synthesis phase, and if you know what the word synthesis means, it means to create. And so here's where we're going to go through DNA synthesis or DNA replication. That's where we're going to copy the original cell's DNA. Why do we need two sets before we go through division? Well, I'm going to take one cell and create two. If I'm creating two new cells, both new cells need a copy of genetic information so that they know how to act, how to function, what to look like. And if they don't have that DNA to tell them that, they're not going to be functional cells. Okay? So if we talk about something like a human cell, we'll start off with 46 chromosomes here. All right, when we go through your S phase, we're going to duplicate that. We're going to create two copies. So theoretically, by the time we get to the end of your synthesis phase, we actually have 92 chromosomes in that cell prepping to divide. Goes through division, we're going to end up with two new cells coming out, each having their own set of 46 chromosomes. Okay. Your gap2 then, we carry out a little bit more normal function. They may do a little bit more last-minute growth. All right, but here we need to then also replicate the organelles. Again, your cells, just as much as those new cells need new sets of DNA, they also need organelles, mitochondria, Golgi bodies, ribosomes in order to carry out their functions. So if we don't duplicate them, then we're not going to be able to ensure that those new cells have every piece of machinery that they need. All right, so that's basically what's happening here in interphase. Now here's a couple other points to make. These cells are going to have checkpoints, okay, and you should mark these on your notes. There's going to be a checkpoint here at the end of gap 1, okay? There's also going to be another checkpoint here at the end of gap 2. And both of those checkpoints are going to be sort of like saying you have to pass Algebra 1 before you can go on to Algebra 2. They're going to be points in time where the cell is going to check to make sure that everything is ready to go, that they have an adequate amount of nutrition in their environment to support new cells, that, especially here at the end of gap 1, are they big enough? All right, are we getting to the point where they're maxing out their efficiency for the surface area to volume that they have, and so we're getting too big? we need to divide in order to reestablish the efficiency of moving materials in and out of their cell. They're also going to look for undamaged DNA. We don't want to send a bunch of mutated cells through cell division just to create new mutated cells. That would be counterproductive. All right. They may also look for signals from other cells. Your cells really want to be right up against another cell. All right. And so when there's a cut, we create a gap between cells. That may be one reason why a neighboring cell may say, hey, I'm supposed to have this neighbor, he's not here anymore, we need to divide to fill that gap back in. Or there is an infection by a virus and it's killing off these cells. Those cells are going to send out SOS signals saying, hey, I'm getting destroyed, we need to make new cells to repair me. Okay, so that's the sort of checkpoints we're going to have here at the end of gap 1 and here at the end of gap 2. Now we also have one more checkpoint that happens during your mitotic phase, and that's going to be right here between metaphase and anaphase, and we'll talk a little bit about that when we get to the actual stages of mitosis later. Okay, but make sure you've got all 3 of those checkpoints drawn on your figure in your notes. Now let's take a look at what a cell actually looks like in interphase, and most of these structures you should already know. So if we were to label these in your notes, and you do have this blank picture there, you should do this. Your nucleus is the big thing here in the center, right? Okay, and that's what most of our focus is going to be on as we talk about the stages of mitosis. That nucleus, remember, is surrounded by a nuclear membrane. That membrane is going to make some challenges. We're going to have to get rid of that membrane in order to move genetic material during mitosis, and we're going to see how that works later. Remember, you've got your nucleolus in the center. That's where we make the RNA to produce your ribosomes. And then finally, if your cell's in interphase, your genetic information is in the form of chromatin. And we want it all spread out in that chromatin versus your chromosomes, because during interphase, we want access to that genetic information. All right, if I have it all tightly bound up and I can't get to it, then I can't express it. I can't go from DNA to RNA to amino acids to create my proteins so that my cell functions. So we want it all spread out so that we can get to the information. It's sort of like telling you that you need to read chapter 6 for homework, but I tell you you're never allowed to open the book. Well, that makes it a lot more challenging if that book is the only place you can get the information to answer the questions, right? So we want you to be able to open the book, or in this case, have the DNA spread out so that we can get to the information. Okay, so interphase is the biggest piece of your cell cycle. Your mitotic phase is going to be the shortest, okay, and that's what's going on right here. And this is really where your cell division happens. Now, here's a distinction I want you to make, okay? Your mitotic phase has 2 subphases within that. We'll talk about the actual mitosis, which also has subphases, and then cytokinesis. Depending on the textbook you read, depending on if you come across a website, depending on even test questions, some of that stuff is written at a level that is not, they try to simplify it, and in simplifying it, they make it incorrect. Okay, so be very careful. I want you to make sure that you make the distinction. If we're talking about going through mitosis, so that's going to include prophase, metaphase, anaphase, and telophase, that's the division of your nucleus. It isn't until cytokinesis, when we divide the cytoplasm plus all the organelles that are in that cytoplasm, that we actually get 2 new cells. So if I were to have a cell that stopped here, never went through cytokinesis, I'm going to have 1 cell at the end of telophase that actually has 2 nuclei, okay, because I've divided the nucleus. But I can't get 2 new cells until I go through cytokinesis. So be careful with that. Mitosis is the division of the nucleus. Cytokinesis is the division of cytoplasm. I don't have 2 new cells until I get to the end of cytokinesis. Something else to keep in mind, we're talking about cells dividing, okay? But your cells are not going to necessarily divide at the same rate. They're not going to go and move through that cell cycle at a consistent pace, all right? It's going to depend on what the body uses those cells for. If you are a skin cell in us, okay, we replace them, your entire body's worth of skin cells, about every 2 weeks. It is one of the most frequently dividing cells because even putting on your shirt this morning is going to have sloshed off some skin cells. It's cells that we brush up against constantly, and so we're constantly losing cells as a result. However, things like your brain cells, your muscle cells, you will reach a certain point in your development where you stop making them. Okay? Your body has divided and created as many brain cells as you're ever going to have by about the age of 3. You will never create new brain cells as a result, and cells that do that end up in what we call a G0 phase, or a no growth phase. They'll go through G1 and they won't ever pass that checkpoint. And so they do what we talk about exiting the cell cycle. And that's what we mean by the G0, which is why things like spinal cord damage and brain damage are such a big deal, because you can't repair them. You can't make new ones. One of the reasons that sports is looking so deeply into concussions, because it is causing swelling of the brain and it is causing damage to cells that you can't replace. Even your muscle cells, you don't ever create new ones after a certain point in development. So how do people make bigger muscles? Well, they're not creating more cells, they're actually tearing the cells that they have. When those cells tear and get repaired, they bulk up. Okay, but they're not actually creating new cells. So keep in mind that not every cell is going to reproduce at the same rate. It's going to depend on what its function is, what the body needs it for, and in some cases, you'll reach a point in development where you never make more. Alright, this is just a reminder, okay? We're making new cells a lot of times for growth and development because those cells can't be infinitely large. Okay, now we also can't be too small because we need them to be able to contain all of the necessary organelles to function. But we can't be so big, depending on the purpose of that cell, because we need to be able to get nutrients in, waste out. Remember the cell size lab that we did, all right, back with cell transport. The bigger that block, the less the color change happened. The smaller the block, you got a color change almost all the way through. All right, and that was there to show you the importance of surface area to volume in order to be efficient. Okay, and this is one of those things those checkpoints are looking for. Alright, so let's sort of recap what we've talked about so far and where we're going from here. Your cells are going to divide for growth and repair during that mitotic phase, and if you're asexually reproducing, then you're also going to use mitosis as a means to reproduce. They're going to make exact copies of the original cell, and what do I mean by exact copies? We're talking genetically speaking. So if I take a parent cell that's prepping to divide in our body, there's going to be 46 chromosomes in that original cell. When we go through the synthesis phase, we're going to duplicate that, so we end up with 92 chromosomes in that cell. We go through cell division, we go through mitosis, and now I'm going to have what we talk about as daughter cells, and each of those daughter cells is going to have 46 chromosomes each. Okay, so they are genetically identical in terms of the number of chromosomes they have and in the information that those chromosomes contain. Keep in mind that mitosis is the division of your nucleus. Cytokinesis is the division of your cytoplasm. So if I stop division at the end of mitosis, I end up with a cell that has 2 nuclei. If I stop division at the end of cytokinesis, that's when I have 2 new separate cells.